Vince is busy right now at home, or he should be at least, finishing his last chapter of his doctoral thesis and preparing for job interviews next week, which is quite exciting. So I'm happy to present this on behalf of my doctoral student, Vince Hustad, who's been working on this for the last 6 years. So the earth tongues, of course, occur in the Ascomycota and have historically been placed in Geoglossaceae, sensu lato. And there's at least 5 different genera that compose the overall group of earth tongues here: the Geoglossum, Laotia, Spathularia, Microglossum, and Cudonia in the overall sense. However, some of the early molecular phylogenetic studies obviously shown that this earth tongue morphology is not monophyletic. Most studies use ribosomal genetic data, ITS, LSU, and SSU, and 3 genera and 4 species were included in this Geoglossum clade in the Hemileucoglossum S. All the red stars on the tree indicate that earth tongue morphology, and what we'll be talking about today is the very bottom clade down here, which is where all the black earth tongues actually occur and appear to be a nice, well-supported monophyletic clade. And then Conrad came along in 2009 with a 6-gene molecular phylogeny that included some protein coding genes, RPB1, RPB2, and translation elongation factor, and showed he created the class Geoglossomycetes and limited it to Trichoglossum, Geoglossum, and Sarcoleotia. These are morphologically distinct taxa. He only included 3 genera and 5 species in their study at the time, although there's at least 50 species total that occur within this group. So now, after several years of work and lots of collaboration around the world, many of you, of course, here help Vince collect specimens, either in the field or send him specimens. He's sequenced 121 collections for the 4 genes shown here, and has done a significant amount of work dividing up the class into these 10 different genera that we show up here in the different colors. So I hope you will agree Vince has done a nice job dividing up the Geoglossomycetes into some workable units. So the remainder of my talk today will basically be talking about these different genera, but first the class overall. Of course, they have this earth tongue morphology. They have a sterile stipe, fertile hymeneum, and a terrestrial habitat. They have these distinctive paraphyses that can either be curved or inflated at the tips. The ascospores can range from filiform and multiseptate to hyaline and single-celled. The entire class is worldwide. The ecology is uncertain, but some species appear to be saprobic, although some may form some mycorrhizal associations at all. And there are sequences in GenBank that have been recovered from soil, as well as root endophyte environmental sequences as well. So we do pick these things up with next-gen sequencing. Of course, the type genus is Geoglossum, described by Persoon in 1794. Vince's gave each of these genera, or most of the genera, some nice little common names in parentheses. He refers to these as the black earth tongues. The ascospores range from hyaline to brown-black and clavate to filiform or multiseptate. The paraphyses are straight or curved, inflated at the ends, and some are darkly pigmented. There's about 27 species, we believe, in the genus now as Vince is currently circumscribing it. Mostly temperate, although there are some in tropical forests and grasslands, and of course the type species is Glabrum. Excuse me. Trichoglossum is what Vince refers to as the hairy earth tongues. Many of you know these because they're separated from Geoglossum based on the presence of setae. Mostly along the stipe. They have these clavate to filiform, multiseptate, darkly colored ascospores. There's about 17 species in the genus. They occur in similar areas as Geoglossum, and the type species is Hirsutum. Brevitrichoglossum is, of course, in parentheses because it's a new genus in preparation. Vince is in the process of describing this right now. It has shorter ascospore lengths and can be differentiated not only molecularly but morphologically from Trichoglossum. It also has shorter setae. 5 species are included and a similar habitat to Trichoglossum and Geoglossum. So the 4th genus is Sabuloglossum, which Vince published a couple years ago. referring to these as the sand earth tongues. They have these stout, rubbery ascomata that are often hollow, hyaline aseptate ascospores, filiform paraphyses that darken at the tips. This is a single species in which we actually took out, we transferred Thumanidium arenarium to this new genus, Sabuloglossum. It has a north temperate distribution that occurs in Europe. We found it both in Europe and North America, and it occurs in moist sand and freshwater habitat as well. And then Vince also described Glutinoglossum in 2013. These are the viscid earth tongues, hence the name Glutinoglossum. They're conspicuously viscid. Of course, unfortunately, not all viscid species belong in this genus, because Geoglossum difforme is viscid as well, but still is a good species of Geoglossum. The asci are narrowly cylindric clavate. Ascospores are initially hyaline, brown at maturity. There's 6 species total so far, and these were all 6 of these species are good molecular species with some morphology to support them, but they were all cryptic species under one name, Geoglossum glutinosum. So a nice example of cryptic speciation being brought into the light with molecular data. They're saprobic on wood and soil. The type species is Glutinoglossum glutinosum. Then there's Hemileucoglossum, the hyaline spored earth tongues. The ascospores are multiseptate, hyaline at maturity, agglutinated paraphyses that are in a conspicuous epithecum. Currently 4 species occur in this group. They're saprobic on wood and soil, and Littorale is the type species. Then we have Leucoglossum, the hyaline spored hairy earth tongues. All right, don't you love these common names? This is great. So the CD are present in the hymenium. It's similar to Trichoglossum in that respect. The hyaline ascospores are not found in Trichoglossum. All of them are darker spored. There's 2 species so far from China and from Europe. These occur in moist grasslands, and Durandii is a type species. And then we have this thing, Sarcoleotia, which has capitate ascocarps with a hymenium distinct from the stipe and not integrating. Hyaline aseptate ascospores, and there's 4 species in this genus. They occur in boggy habitats in Europe, North America, and temperate South America, and Niagara is a type species. And then we have Nothomitra, a relatively newer genus with a capitate hymenium, which is distinct from the type. The ascospores are hyaline to lightly colored, single-celled, and multiglutinate when young, becoming septate when over mature. A small genus with only 3 species in north temperate latitudes, and synonymy is the type species. And then last but not least, we have Masoglossum, in which the ascomata are similar to those found in Geoglossum. However, the ascospores in this case are single-celled, and they can be warty or smooth. The stipe hyphae is similar to Thumanidium atropurpureum, so there is a possible link here to the Geoglossomycetes, since this genus occurs at the base of the tree. There's 2 species known from Bhutan and Sweden, so quite an interesting distribution there. We believe it may be saprobic in heavily disturbed habitats, and the type species of Vericosporum. So I'll just go back to this phylogenetic tree again, because the nice thing that Vince worked out was there seems to be some evidence of ascospore evolution. If you look at the base of the tree, we have these hyaline aseptate ascospores. And as you move to the more derived genera at the top of the tree, they become brown and multiseptate. So there's some evidence of some interesting ascospore evolution here as you move throughout the group. All right, so our knowledge of the Geoglossomycetes is obviously greatly increased thanks to the work of Vince. Much greater diversity occurs within this class than was ever previously known. I've tried to show you some of the ecology which may play a role in the sympatric speciation. It's a nice model example that even small, well-studied groups of fungi, such as the Geoglossomycetes, can have an interesting story to tell. So last but not least, Vince, of course, would love to thank all of his funding sources, but also especially his collectors and all the assistance that he got. Paul Cannon was instrumental in helping him collect throughout England and Scotland. Peter Johnson helped out in New Zealand. And I'd just like to take a minute to thank Don Feaster. When we started out on this study and Vince was coming to me for the first year of his work, I spent a couple $100 to bring Don Feaster down to the Great Smoky Mountains with Vince. And although it cost me a couple $100, the experience was priceless to be able to go out and collect with someone like Don Feaster for a week and learn about Discomycetes. And at the end of that 1 week experience, Don shared some of his pearls of wisdom because Vince didn't know exactly what he wanted to work on. He just knew he wanted to be the next Don Feaster of the world and work on Discomycetes. So Don suggested he pick up the Geoglossomycetes, and we've been forever grateful for that, Don. Thank you very much. And with that, I'm more than happy to try to answer questions, but feel free to email Vince. We were going to try to Skype him in to answer questions, but you know, that doesn't work. It's 3 in the morning and he's still asleep, I'm sure. So thank you very much.